All right, peace be upon you all, and welcome to another episode of Seeing God in Biology with Dr. Fuz Rana. And today we're going to talk about genetic code. Fuz, how are you doing today?
Ahmed, I'm doing great. How about you?
All is well. So genetic code seems like a fun topic, and I'm sure you're going to tell us some very interesting information there. And does code refer to like computer code or like as if it's like programming?
Yeah, well, here it's more like a set of rules that
Okay.
really help us to, in a sense, make sense or define the information that's stored in DNA as well as the information that's functionally expressed in proteins. So it's necessary in a sense to decode the information. It would be similar to the Rosetta Stone, if you will. that
Aha.
that you have these hieroglyphics that make no sense at all. But once you understand the Rosetta code, or once the Rosetta Stone was found and decoding of the hieroglyphics took place, now that information was essentially laid bare, and that's the same thing with regard to the genetic code.
All right. So let's see what you have for us in the presentation then.
Sure thing,
Interesting.
and I'm really looking forward to your thoughts and your comments because I think this is right up your alley. And
Yeah.
you know, we've been talking quite a bit lately about the central dogma of molecular biology, and the genetic code factors centrally, really, into the central dogma.
Yeah.
And just as a very quick review, the central dogma basically points out that there is information harbored in DNA, and this information really are the instruction manual that uh, really allows the cell to build itself, and then kind of directs the operation of all the cellular components in such a way that the cell is able to grow and to divide.
Yeah.
And interact with its environment. And the information that's stored in DNA, which is digital information, consists of discrete units that are called genes, where each gene encodes a biomolecule called a protein.
Mm-hmm.
And proteins are really the workhorse molecules in the cell that are carrying out all the different activities, playing a role in forming the different cellular structures. And the way the information moves from DNA into proteins is through the central dogma, where that information in DNA is copied through a process called transcription, where each gene that is copied now resides, or that information resides in a molecule called RNA, which is very similar structurally to DNA. So both DNA and RNA are nucleotides, polynucleotides. And then that information in RNA is read at the ribosome, or read at ribosomes to produce proteins. And we talked about the process of translation in a
Yeah.
previous episode as well. And so we see again this flow of information, but notice that what's happening is the information is changing formats. It's going
Mm.
from a polynucleotide format to a polyamino acid format. It's going
Yeah.
from nucleotides to amino acids, and there has to be something that mediates that change in format. And that, in
Mm-hmm.
effect, is the genetic code, which ultimately consists of a set of rules that, let's see here, here we go. It consists of a set of rules that relate the information in DNA and RNA to the information in proteins. And so this is a typical diagram that biochemists will work with that
Uh-huh.
defines the genetic code, that shows how, again, nucleotide sequences translate into amino acids. And
Mm-hmm.
what we note is that in DNA and RNA, there are 4 coding units, 4 different nucleotides, the genetic letters, abbreviated A, G, C, and T for DNA. Yet in proteins, there are 20 amino acids that are specified by the genetic code. And so, how do you go from 4 nucleotides to 20 amino acids? And so,
Mm-hmm.
obviously, there can't be a one-to-one relationship between nucleotide sequence and amino acid sequence.
Yeah.
Right. And if you say, well, what about having kind of a coding pair where you have two nucleotides? Well, that's 2 to the 4, or essentially 16 possible coding units. That still isn't enough. So the smallest number of coding units that you could have to correspond to the 20 amino acids would be 4 to the third. Sorry, check that, 4 to the third power, which would be, so I misspoke earlier. It's not 2 to the fourth power. It's 4 to the second.
4 to the 2. Yeah.
Yeah. So it would be 4 to the third power, which would be 64 coding units. So the genetic code consists of what are called codons or coding triplets, where it's combinations of 3 nucleotides that correspond to each amino acid. And in fact, the scientist that deciphered the genetic code won the Nobel Prize for that work, because this is, in a sense, the heart of biochemistry, I would argue, as a biochemist, that this set of rules here defines the information that you see in the cell. It gives meaning to the information in the nucleotide sequences of DNA and RNA, and then it shows how that information then can be functionally expressed in the form of proteins. So this is essentially the genetic code. Now, because there are 64 possible coding units that correspond to 20 amino acids, it means that there are some amino acids that are specified by more than one coding unit. This is called the degeneracy of the genetic code, or you could think of it as the genetic code's redundancy.
Okay.
And so, again, there are these 3 nucleotides that correspond to each amino acid. These are called, again, codons or coding triplets. And this is another diagram that kind of helps to explain that relationship again, where if you look at, this is just one of the strands of, well, check that, that's actually a strand of RNA being depicted. So this would be a copy of the information found in DNA. It's existing now in the form of an RNA molecule. And as you go through from top to bottom of the RNA molecule, you'll see, again, 3 units, 3 nucleotides that form, again, a coding unit, where in RNA, thymine is being replaced by uracil. And so you can see the different codons. So again, it's a coding triplet. Now, what we see here is that the genetic code is read sequentially from start to stop. So there's no punctuation, there's no interruption in the sequence of coding units. It again goes from start to stop, but you do actually have start and stop codons. These are codons that specifically tell the cell's machinery, this is where to begin reading the information in RNA as we begin to translate it to produce proteins, and that this is where essentially the sequence stops. And the start sequence is much more important than the stop sequence, or the stop codon, because the RNA is eventually going to run out, run off the ribosome.
It's going to finish anyway.
Yeah, exactly. So the stop codon isn't as important, but the start codon is absolutely critical in terms of getting the precise positioning of the RNA molecule on the ribosome. Because you can imagine, if you get a mispositioning of the RNA molecule on the ribosome, you now are going to be offset by 1 or 2 nucleotides, which means you're going to actually have an alternative reading frame, which will give you the wrong protein, if that makes any sense.
Yeah, absolutely. Can I interrupt you here, or maybe we
of course.
just go? So actually, this is a very nice slide, because while you're talking, it's definitely making a similarity to how information is coded, for example, in computers. So, for example, the typical systems today use what is called an ASCII code. Okay, and ASCII stands for, I guess, American something, American Standard for Information Coding or something. And how it works is that you have, instead of 4 to the power 3, instead of the nucleotides, which can take one of 4 different values, you have bits which take one of 2 values, and then you have the alphabet. So you have letters, and you have the letters in the uppercase, and you have the letters in the lowercase, and you have the plus and the minus and the numbers and the rest of it. So people have, chosen to use a code of 8 bits. So you would have 2 to the power 8, which is typically called 1 byte, which can code for 256. And then let's imagine this. Let's imagine that somebody finds, 10,000 years from now, finds a paper where, you had 010101, which would be, instead of GT, A and C, and then somehow this brilliant guy finds the Rosetta Stone, and he finds out that if the character code is reading so and so, it means letter A, and if it's so and so, it means letter B, etc., etc. And he figures out that it's every 8 are what constitute a character. And then he finds this scroll full of bits of zeros and ones, and he uses the decipher Rosetta Stone, and voila, he has a Shakespeare or something. So it's a great story or a great poem, and then somebody would come to him and tell him, guess what? This is just random. It was completely random zeros and ones. And because there was no intelligent life 10,000 years ago. So this must have, either came by mere chance or evolved over a period of time, over a computer that was just crunching numbers, and there was a selection mechanism of some sort. And hence it produces this novel. But it couldn't have been produced by a person like Shakespeare. And I guess everybody will say this is totally absurd. So, I mean, the stunning resemblance here between bits, in this case, it's not a bit in terms of a 0 or a 1. It's the 1 bit of the genetic code can take 4 possibilities. Well, let's call them bits here, just for the analogy. And then you have a collection of them that is consistently of length 3. And then, because, if this guy who was translating the zeros and the ones couldn't know exactly how to start or exactly where to end, and there was a little piece of the paper that was cut off, and he just has 3 shifts or 1 shift of bit or 3 shifts of bit, then the whole thing would be nonsense. So we do the same even when you're talking about the start and the stop in computer communication. We always have this one combination that will be not used during the transmission of the text. Okay, it can be a set of trailing zeros or a set of specific sequence that we will make sure that it doesn't happen in the normal transmission. And, it's like when those pilots or those people in the military are talking, and then he says, over and out at the beginning, or something like Roger or something. And you typically don't try to use the word Roger within the dialogue. So if somebody's called Roger, you'd better not have him here, because then this and they would do these comics about that, who is at first place, and there is somebody in the team called who, and and it's all of a mess. So even the idea of having specific code words used for starting or stopping, to be sure that you are starting at the right bit, and then you can count 8 bits, and then you go, and then instead of the letters, the characters, which is the alphanumeric letters, then you have the letters here that are representing proteins or amino acids in this case, and then the sentence or the novel, the poem would be the protein, or actually the collection of proteins that come out of reading the whole scroll, which is in this case the genetic strand or the chromosome or the whole genome. And then at the end of the day, each one of them is a verse in the poem. And at the end of the day, you have a complete poem, or you have a complete novel, which is a human being or a rabbit or a flower. And when you look at that, I'm really wondering how somebody who understands information systems or understands biology, or even has the very least rational perception of life, would resolve to anything other than that this is a work of an intelligent designer who knows exactly what he was doing. So I hope that was not a long, a prolonged thought, but the analogy is totally stunning, definitely.
Yeah, well, I'm first of all very grateful for your input there, because, to me, it is absolutely astounding that, we know from common experience that information comes from a mind. And we know that if you see it, that information that's encoded, that code, that set of rules must come from a mind. But what really adds to the eeriness of it all is the fact that structurally the way the genetic code is put together resembles, again, the ASCII system. And what's interesting is that there is this efficiency to the genetic code that, from an evolutionary perspective, why not have a coding, what would you call it? Not coding triplet, coding quadrillet or quintuplet, right? Yeah. But those would be essentially unnecessary. That would be excessive. So what you see with the genetic code is this highly efficient system where you have just enough complexity to accommodate the 20 amino acids, but not excessive complexity that would be superfluous. And so there's an elegance to the genetic code in that sense as well.
Absolutely.
Okay, well, let's kind of press on here.
Yeah.
Now, something else that really is significant about the genetic code is what biochemists call its degeneracy, or what we could think of as its redundancy. That's very much part of the genetic code, because there's excess coding units compared to the number of amino acids. And so, for example, it's maybe difficult to see here in this diagram, but if you look at the coding unit UUU and then the coding unit UUC, they both code for the amino acid phenylalanine. There's actually 6 codons that code for the amino acid leucine.
Mm-hmm.
And so, some of the amino acids only have a single coding unit that specify it. Others have 2, some have 3, some have 4, and so on and so forth. So there is a redundancy to the genetic code that, again, as we'll see in a minute, is absolutely critical to its design. This is another way you could depict the genetic code, where the 5' position would be one of the genetic letters in the inner circle, A, G, C, or U. The second position would be in the second ring, either U, C, A, G, and then the third position would be, again, in the outer ring, where then the different amino acids are specified. And if you look at it, in this way, you can, again, see that some amino acids are specified by, more than one coding unit. Some are specified only by a single coding unit. And so the width, you have this little slice of pie for the ring, and the larger the slice of the pie, the more codons that are specifying a particular amino acid. So this is another way of looking at the genetic code.
Yeah.
Now, one of the things that is really interesting about the genetic code is that that again, you see this this redundancy in the genetic code, and you see these different assignments. The genetic code is in effect universal. The genetic code is pretty much found in every living organism. And so you could think of it in that sense as universal. It's also sometimes called the canonical genetic code. Now, it is true that there are genetic codes that deviate from the universal genetic code that are found in nature, mitochondria, for example, and some other organisms have slight variations of the genetic code. Mitochondria have a genome, and usually the mitochondrial genetic code is slightly different than the universal genetic code. And so this is just a diagram showing that difference, and it usually involves one or two positions in the genetic code that are slightly different, and they almost always involve codons that are stop codons, where what was a stop codon in the universal code will specify a particular amino acid in the deviant of the genetic code. And so you could explain this through a limited type of evolution, through a limited microevolutionary change, where again a stop codon is reassigned as a codon that will specify an amino acid. And this typically isn't devastating, because if you're already towards the end of that RNA molecule anyway, changing the stop codon to an amino acid is going to have very minimal effect. And or if you do see a change, it usually is in what's called a low frequency codon, where it's a codon that just simply isn't used very much in the genetic code. And so the reason why this is significant is because typically you would argue that the genetic code really can't evolve in large scale terms, because if you start changing a codon, right, where one codon now is coding for another amino acid, that effect is going to reverberate through the entire organism. Every protein is now going to be impacted by that change, and that will ultimately be devastating to the organism. In fact, Francis Crick, who played a role in discovering the structure of DNA, also played a role in essentially characterizing the genetic code, and made the argument that the genetic code can't evolve, that it must be, from an evolutionary perspective, a frozen accident that was instantiated in time, because once the code is in place, changing the code is going to be devastating to the organism, unless you're dealing with low frequency codons, that occur very rarely, or if they are stop codons. That's typically going to be the only place where you could really tolerate any kind of code evolution. And so when you do see deviants of the genetic code, these are really deviants from the universal genetic code, and it's a limited number of changes. It's not a large scale change. So a lot of times I'll hear skeptics say, well, the genetic code is not universal. Well, that's not really true. It is universal. You're just dealing with rare deviants that can be readily explained through microevolutionary changes. And in fact, in the mitochondria, some of the changes that you see in the genetic code are essentially replacing amino acids with methionine, which is an amino acid that can actually help absorb oxidative damage to proteins through a redox cycle that involves the R group of methionine that can undergo oxidation and reduction. So there's actually a rationale or a logic to the deviant codes that you see that makes sense why that change would actually take place. Okay, so this is a point that we've already kind of elaborated on, but the fact that we see a genetic code at all, to me, right away suggests that there's a mind. Again, because we know from experience that codes are things that are constructed from a mind, that they're oftentimes there's an arbitrariness to a code where it's just simply a set of rules that somebody devises, and then that person gets agreement with other people that this is going to be the code. This is how we're going to make sense of the information that is, digital, that digital information is through the use of the code. That really, again, gives meaning to that information. And again, as you're saying, there's this eerie resemblance between the structure of the genetic code and again, ASCII coding.
By the way, one of the ways that information is coded and then protected from misrepresentation is through something called cyclic redundancy check. So it's funny to me that actually even information systems use the same concept of redundancy to, in this case, to detect actually if the information has changed or corrupted during communication. But some other systems would use just mere redundancy. So it will just store the same piece of information, for example, on 3 drives, for example. And then you will, if one of them is tampered with, then you can have other copies. Some very recent systems, for example, like the system that codes for cryptocurrency, for example, for Bitcoins, for example, you will have the same piece of information on maybe 10,000 servers, for example. And that's why it's very difficult to fraudulently create a Bitcoin, because if you say you own it, you need to convince 10,000 servers of this misinformation. And when one of them would claim something, at least half of the others have to agree with it to vouch for the truthfulness of the information. And this is how blockchain works. So at the end of the day, this, let's say, degeneracy or having to be inefficient in storage is actually something that engineers actually use to protect information from being misrepresented or corrupted or changed during transmission, or even maliciously changed for fraud or for other stuff. And again, a hallmark of design and a hallmark of a wisdom that is behind the whole system to make sure that there is integrity and there is consistency all over it, actually. And actually, even the very fact that this is digital. Digital communication is by definition inefficient because, for example, if I give you a piece of paper and I write like 8 blocks on it where you can only write 8 characters, it limits what you can write on the same space versus if you would, you can do any scribble. So if you can just scribble continuous data, you can actually use the same space to write more. But the issue is that digital information is way less prone to distortion. So if I can only be saying yes or no, then whatever I say, you can either translate into a yes or a no. There cannot be a mo, for example, or a yo. If you hear a yo, it has to be a yes, maybe. And if it's maybe a yo would be a trick because it has the yeah and has the o. But any other thing would be either closer to the no or to the yes, or to the 1 or to the 0. So digital systems, the fact that a system that looks very analog in nature, in its representation, natural systems are typically analog systems. So you'll have sunlight, for example, and you can have very dim light or very bright light. The room can be very dim or very bright, and everything in between. Red can be very red or can be light red or can be very deep red, and there is almost everything in between, okay? But then when you look into the very design of the fundamentals of biology and you find that it is digitally coded, it is it is so stunning, because the fundamental system that codes for the information, intrinsically in it has the idea that this place can be one of four things. It cannot just be any chemical. And then error correction and detection and damage repair can guess that this has to be one of four things. It's in A, C, G, or T. It cannot be anything. And then it can have the other redundant strand, like the RAID systems in hard disk storage. And then there is all other error correction stuff. And then there is the translation and everything. A huge resemblance to, at the end of the day, when humans were thinking of a way to systematically and robustly save and communicate information, they eventually resorted to digital systems because they are resilient to noise and to distortion and to corruption. And they resorted to redundancy because it gives you all the robustness against fraud and a malicious change or accidental change of the data. So I hope that reconciles with what you have in mind.
Oh, it does, and yeah. It's a wonderful segue into the next point that I want to make.
Oh,
because it's interesting to me that so many biologists will look at redundancy and see that as being superfluous, right? And they'll argue, well, redundancy is evidence that that system was cobbled together by evolution, because you just have this unnecessary duplication. But as you're pointing out, redundancy is usually a technique used to ensure the robustness of the system. And in fact, with respect to the redundancy in the genetic code, it's very tempting to say, well, the minimum number of coding units that or the minimum size for coding units in the genetic code must be 3, because you have 20 amino acids. And so therefore the extra coding units were just simply,
not necessary,
simply just, were assigned, through random processes to be duplicates corresponding to, the same amino acid. But it turns out that the more people have probed the redundancy of the genetic code, the more that they found something really quite remarkable, and that the genetic code seems to be highly optimal to withstand error that could occur during data transmission.
Wow.
During the translation process, that the genetic code is exquisitely optimized.
Wow.
And in fact, Simon Conway Morris, who's an evolutionary biologist, also a theist, said that the genetic code displays eerie perfection and startling evidence of optimization. And to appreciate that, we need to think about the types of errors that can occur.
Can I make a guess? Can I make a guess, and you tell me if the guess turns out correct?
Yes.
And I'm really making a guess here.
Okay. Yes.
Really
go ahead. It'll be a lot of fun.
Okay. So I hope this is a correct guess. So if I'm designing this, I'm not, obviously, but as the creator of the universe, maybe I will cause the codes that are due to their chemical nature are more prone to biochemical change, and I will give them more redundancy. I will make more codons represent them than the ones that are less prone. So I will put more redundancy where more error is possible. This is so, if this I don't know, a specific amino acid is represented by that one codon, but this codon is made of those chemicals where, if it changes, it will send us to the moon, then maybe the possibilities of change, I will make them another redundant representation of the same protein. I hope my guess works.
Yeah, you're very, very close, if not on target.
Okay.
Well, but this is a very important point, because what it's saying is that as a human designer, there's a rationale for how you would structure redundancy. It's not just haphazard. It's
Absolutely.
Well thought out. And this is and it's thought out in a way that you're going to minimize error. And this is exactly how the redundancy in the genetic code works. And just to illustrate that, here's a slide showing essentially a very small piece of a protein and the codons that code for it. Now, if you have a substitution where one of the nucleotides is replaced with another one, in this diagram, a C is replaced by a G, that coding unit is now going to specify, instead of proline, arginine, right? And so that's a big change in amino acid. That's a big amino acid change that could potentially disrupt the protein structure. You can also have insertions and deletions where either one or two nucleotides are inserted into the DNA, which now would cause what's called a frame shift mutation, where you're now literally changing from that point in the protein, all the other amino acids now are going to be changed because of that frame shift. Frame shift mutations are really bad news. Substitution mutations are bad. Frame shift mutations, whether they're through insertions and deletions, are really bad. And there can be a single insertion or two insertions. Now, if you have three insertions, you will actually cause a frame shift. Actually, you won't cause a frame shift. You'll just cause an amino acid to be inserted into the sequence, but all the other amino acids will be the same. But the likelihood of three insertions is very low. The likelihood of one or two is very high, or deletions, one or two is much more likely than three. So anyway, two types of mutations we're dealing with: substitution mutations and deletion and frame shift mutations. Sorry. Now, this is showing again the same kind of effect using human language as the illustration, as opposed to a DNA sequence. So if, for example, you have a G that's replaced with a T, that's a point mutation or a substitution mutation, the sentence changes, but it's not horrific, right? The man saw the dog hit the can, and now it's the man saw the dot hit the can. The meaning has changed, but it's and significantly, actually, but nevertheless, you still have most of that sentence intact. If you have a deletion, then they would say the man saw the hit the can, right? So the dog is removed, or dot is removed. If you have an insertion, then you could have, like in this case, the man saw the fat dog hit the can, right? So that gives you an idea of how these different types of mutations can impact the meaning of proteins, just like they would impact the meaning of a sentence. Now, it turns out that if you examine the genetic code, and this isn't the best diagram, and I apologize for that, but if you look at the genetic code, what's interesting, and this is to your point that you made, that most of the changes in the codon from a substitution mutation are going to generate a new codon that either encodes the same amino acid or an amino acid that has very similar physical chemical properties. So if we start in the upper corner, U, if you change the last U, the U in the 3' position, to a C, you get phenylalanine. If it's an A or a G, you get leucine, which is very similar to phenylalanine in terms of its physical chemical properties. Leucine, again, has 6 coding units. But and then isoleucine is very similar to leucine. And so that valine is also a hydrophobic amino acid similar to isoleucine. So as you start working your way through, just through qualitative inspection, you begin to see that any kind of substitution mutation is going to generate either the same amino acid or one very similar that's going to have minimal disruptive effect on the structure of the protein.
Wow. Now, actually, this is very close to what I was hoping, but it's actually even better, because what you're saying is that, so for example, for this one in the upper left corner, if the U becomes a C, it's still the same thing. But if it becomes an A or a G, then even this amino acid substitution maybe will cause a little bit dysfunctional protein, but maybe the function is not that far away. So it can be deficient, but maybe not completely not functioning.
Right.
And actually, this is much better than our ASCII codes. So if I map this to an ASCII code, for example, imagine that the designer of the ASCII code will not code A, B, C, D in sequence, because the ASCII code of A and then B just comes after, and then C just comes after. But imagine if the ASCII code of C has the ASCII code of K right after it, and maybe G right before. So if one bit maybe changes from C, it becomes a K. Or if it's one bit less, then it becomes a G. So if somebody's reading, cake, and he finds it K-A-K-E, he would probably guess that this was originally cake.
Yes.
Or if it's not like cake or something, if it becomes a G. Or let's consider, for example, that I would design it so that the ASCII codes of the vowels come after each other. Yes. And it will be the A, and then the I, and then the O, and then the U, for example, and maybe EI, and then maybe the Y is just right next to the I or something. So an I can be a Y or an E, for example. So, and this way, I would re-engineer the ASCII code to the extent that even if after all my error correction system, something slips, then the reader can guess what I meant.
Yes.
But this is not even in our system, so this is even much better.
Yeah. Well, what's interesting is that there have been biochemists that have now that have quantified the error minimization capacity. And I won't get into the details how they went about doing it. But one particular study, the researchers generated 1 million random genetic codes where they just randomly assigned the codons to amino acids. And they had 1 million random codes, and they compared the error minimization capacity to the code found in nature, where the lower the number on the x-axis, the greater the error minimization is.
Wow.
The larger the number, the higher the error minimum. So in other words, you want the lowest possible number, right?
I'm saying wow because I see the natural code.
Right, and so they
Right
generate
out.
this bell-shaped curve, right? And the code in nature is in effect a statistical outlier. Now they looked at 1 million codes, and they could have looked at 10 to the 18 codes that have the same type of redundancy, and then there's 10 to the 80 codes that have that could be generated in principle. But because you get this bell-shaped curve, even though you're only looking at 1 million codes, you would assume that all the other codes are for the most part going to fall within that distribution. And so the title of the article was the genetic code 1 in a million, right? And really it's probably even much greater in terms of its error minimization capacity. So in other words, the code in nature is this statistical outlier in terms of its error minimization capacity.
Wow.
So it's not just simply...
And this only considers the mathematical aspect. It doesn't even consider the biochemical aspect of the compounds we're talking about, right? The fact that the phenylalanine and the next one and the next one and the next one would really be good relatives if it moves from one to the other. It just considers the absolute error issue.
No, it actually, in this analysis, it included the biochemical properties.
Wow. Okay, so even including this, we're still at the minimum.
Yes. And then more recently, a team of researchers did a similar analysis looking at frame shift mutations. And this is what they say in their article. We conclude that the standard genetic code is not only very efficient in minimizing the consequences of mistranslations, but rather optimized in amino acid polarity conservation for all three effects of code alteration, namely translational errors, point and frame shift mutations. In other words, our result demonstrates that the standard genetic code appears to be more than just 1 in a million. Wow. So in other words, it's not just simply point mutations, it's also frame shift mutations where you see this kind of optimization.
Amazing.
And in addition to that, in addition to the genetic code, there are other codes that are imposed on top of the genetic code. So within a stretch of DNA, you have multiple overlapping codes that are doing different things. So some of those codes are coding for proteins, others are coding for histone binding, which is necessary for creating the three-dimensional compact structure of DNA in the nucleus. There are regulatory proteins that are binding to DNA, splice signaling, secondary RNA secondary structures. So there's a whole ensemble of other codes superimposed upon the genetic code. And it turns out that the genetic code is also optimized to harbor overlapping codes as well.
Wow.
And so what you're looking at is not only the fact that you've got a code that is designed identically to the way we would design something like the ASCII code, where there is redundancy in the code that would be, again, the type of redundancy we would incorporate into information systems, but that redundancy is exquisitely optimized. So it might be one thing, Ahmed, for you as an engineer to design redundancy in strategic places in your information system, but to say that it's optimized compared to all possible ways in which you could incorporate redundancy would be almost impossible to truly achieve. Absolutely. Even with the best engineers in the world, with all due respect to people that are very talented engineers. Yeah. But what you're looking at in the genetic code is this optimization that is extreme, and it's a multidimensional optimization, right?
Yeah.
It's optimized for all kinds of other factors that information in DNA is intended to do. And
you said you said they tried to create the bell shape 10 to the power something crazy number, and yes, you need a computer and a program to do the calculation. It's impossible to do it on a piece of paper, set aside just by trial and error, which will take, I don't know, infinite number of universes maybe.
Right. Well, exactly. And so this is the next point that I want to make.
Oh, I'm sorry.
Because, again, what you see is that, what Francis Crick argued is that the genetic code must be a frozen accident, as I mentioned earlier, because any kind of evolution of the code, other than introducing small changes with stop codons or low frequency codons, would be lethal. So the code, according to Crick, can't evolve. Well, how then do you explain a genetic code that's exquisitely optimized? Well, from an evolutionary perspective, it must be natural selection. But how's natural selection going to form, explore coding space when code evolution becomes lethal? Yeah. Let alone how much time would it take, right? This was to your point, right?
Absolutely.
That, if you have, let's say, 10 to the 70 codes, and you look through essentially, and you've got a universe that's whatever, 13 billion years old, you've got to go through 10 to the 15 codes per second.
Wow.
Right? To somehow discover this optimal genetic code that we see. Right? And even if you have, you have 10 to the 15 seconds, so you'd have to go through 10 to the 55 codes per second. I'm sorry, I misspoke, to find the code in nature.
If you divide.
Exactly. Now, let's say you had 1 trillion organisms that were all evolving at the same time. That still what drops it from 10 to the 58 to 10 to the 43?
Yeah,
codes per second.
It doesn't make a difference.
Right. So you don't have enough time, and
you're not even close to having enough time. It's totally ridiculous.
Right, and the famous evolutionary biologist Manfred Egan, in a paper published years ago, basically showed that the genetic code seems to have originated at the time of the origin of life. So you've got, the code can't evolve. Even if it could evolve, there's not enough time to find this highly optimal code that seems to be practically unique. And by the way, when we look at the natural history of life, the origin of the genetic code coincides with the origin of life. So you really are forced into a framework where, it ultimately is designed, right?
Absolutely.
Now, we've been going on for about 50 minutes, but there's one other point that I want to make that to me is just,
Yeah,
if this isn't mind-blowing enough, right?
Yeah, it is actually.
The source of the redundancy in the genetic code is due to something called the wobble hypothesis.
Okay.
Now, so the amino acids that are brought to the ribosome are bound to transfer RNA molecules. We talked about that, right? And that each amino acid has a corresponding transfer RNA molecule, right? And the on the ribosome, the messenger RNA is there, and there is the anti-codon of the transfer RNA that recognizes the codon of the messenger RNA.
Fits with it.
Right. Yeah. And it uses what's called Watson-Crick base pairing rules, the same rules that are used in DNA, right, to hold the two DNA strands together.
Yeah, the parity thing, it relates to the parity thing that we talked about. Yeah.
Yes, exactly.
Yeah.
So if the messenger RNA is going 5' to 3', the anti-codon of transfer RNA is going 3', 5', where on the transfer RNA, the number 1 and number 2 positions going 3' to 5' adhere to Watson-Crick base pairing rules, but the third position has some freedom. This is called the wobble base, where you can have the same, it no longer conforms to Watson-Crick base pairing rules. There's some flexibility, some slop, if you will, and that creates the redundancy. So the base pairing rules are flexible in the wobble position.
Can you repeat that one again? Just the last position piece.
Yeah, the last position,
Yeah.
which would be the number 3 position in the anti-codon,
yeah,
is called the wobble base. In other words, there's flexibility. So the G doesn't always have to recognize a C. The G can actually recognize maybe a T, or maybe and still you still would get that the position.
It will still bind. It still bind. Yeah.
Yeah. So there's some slop in the system. And so the wobble position, which is the third base of the messenger RNA codon, and its corresponding tRNA position, and there's flexibility. And it turns out that, so this illustrates this. So let's say you've got in the upper left-hand corner the transfer RNA that's bringing leucine, and normally it would be a GAG pairing to a CUC on the messenger RNA. In the wobble position, that GAG would also recognize CUU, where the C in the U, you would still have enough positional force with those two positions to get the right amino acid there. So you get that wobble base creates some slop. Now, sometimes in transfer RNA molecules in that position, that third position, you have a nucleotide that doesn't occur in DNA or messenger RNA called endosine. And the endosine can actually base pair with U, C, and A.
Okay.
And so on that wobble position, sometimes what will be incorporated will be a nucleobase that has even greater flexibility than the Watson-Crick bases would have.
Okay. All right. And how does this endosine come into play anyway?
It's actually incorporated into the transfer RNA after it's transcribed. So it'd be post-transcriptional modification, where there's biochemical machines that would actually chemically alter the nucleotide in that position to produce adenosine.
Okay.
Or endosine, sorry. And so there's actually a set of wobble rules that have been developed where G on the anti-codon can pair with U or C, a C can pair only with G, and A can only pair with U, but a U can pair with A or G, and then an I can pair with U, C, and A. Now, what this means, in other words, this is explaining the redundancy, at least in large measure. The redundancy is actually due to this wobble base, right? And that means that you can have 31 transfer RNAs that can actually read 61 codons. So this is where, and if you start looking at the redundancy It's actually arising out of the wobble bases, or
Yeah.
not all of it, but a significant portion of it. And
Yeah.
so again, the redundancy in the genetic code, which accounts for the optimal nature of the genetic code, is actually specified by the chemical structure of that wobble base, right?
Uh-huh.
All right. And so in
Wow.
other words, it's almost as if the law that the
It's a
redundancy,
conspiracy with the laws of physics and
it's
chemistry.
prescribed by the laws of nature, right? So
Yeah,
the laws of
amazing.
nature are prescribing the Watson-Crick base pairing, and it's prescribing, in a sense, the wobble base pairing rules, which happen to give you redundancy that is just the
Amazing.
just right redundancy to create this optimal genetic code.
Yeah, yeah, I get it. Yeah, so,
This is
yeah,
crazy, right?
totally amazing. Yes,
So,
yes,
so when
absolutely.
people ask me,
Hmm.
as a biochemist, what do you think is the most compelling? If there's one piece of evidence for design that you could cite, and only one, what would it be? And to me, it would be the optimal nature of the genetic code, because wrapped into that is just this all these design tentacles that come off of it's like
Hmm.
the head of the octopus, right, is
Yeah.
the optimization of the genetic code, and there's all these design tentacles that come off of that optimization.
Actually, it something just popped in my head that there is a reverse conspiracy also embedded in this one conspiracy. That if you're trying to design this, like, let's say, as a human or an alien or something, you need to be aware of each and every law of physics, chemistry, and biochemistry to the extent that you will pick the exact code, the exact chemicals that represent the code, and the exact mechanisms that will execute it. And if you don't really pick the right ones in a streak, one law of chemistry or physics will actually act against you to the extent that what you create will never be self-sustainable. It will die away or halt or degenerate so fast or get wiped out one way or the other, because otherwise, if you implement redundancy but the redundancy is not consistent with those wobble rules, then it leads to nothing. It just actually leads just to degeneration without the redundancy positive effect. So I'm not sure if that was quite explanatory, if I meant
No,
what I meant.
I perfectly understand exactly what you're saying.
Yeah.
It's yeah, it's literally a house of cards that if even one little, one part of that house of cards isn't even,
Yeah.
if the card isn't even quite the right size, it's
Yeah.
just all going to collapse.
It's like a key and a lock, and you need all the pins of the key to be exactly like all the dents of the lock. Otherwise, it will not click.
Yes.
So you have to get every single pin, every single digit on the digital lock or on the analog key. Otherwise, it will not fit in. It will not unlock the door. That's that's how it is, and this can only be by design. That one was really mind blowing. I have to agree.
Yeah.
Yeah,
Yeah.
yeah,
Yeah. So, so it's just hard to wrap your mind around, what it really means to say that the genetic code is optimized because of the redundancy that seems to be fundamentally, again, prescribed by the laws of nature. It's, it's crazy. And the chance of hitting upon that genetic code is virtually impossible through evolutionary means.
absolutely. And the standing, let's say, testimony to this is that this has been life on Earth for billions of years, and we're still here.
Yes.
And we can have this conversation, and it's very resilient, and apparently to us it's self-sustainable, which is nothing like any system that we can build, no matter how brilliant we are. It has to be externally maintained all the time. But this one, at least apparently to us, because I believe that God is intervening all the time, but at least apparently to us, it's self-sustainable, which is quite amazing.
Yes.
It is the standing testimony that this can only be here by a perfect fit between what is engineered and the fundamental laws of physics, chemistry, biochemistry, and you name it. Yeah, information theory, everything.
Yeah, and what's interesting to me is that there are some biochemists that see this and recognize the fact that this just seems to be really very eerie, and there are other biochemists that are doing everything they can to try to make the case that the genetic code isn't optimal, right?
Wow.
And so there's a bit of a debate going on in the scientific literature, but as that debate's happening, there's more and more new insights to the optimization. There's other examples of optimization that I didn't even. What's that?
The more you try to prove it's not optimal, the more it pops up as being even more optimal than you initially thought.
Yeah. Well, there's other aspects of the optimality that I didn't even bring into the conversation.
Wow.
So it's the design is amazing. So
amazing. So as we say in Arabic, Subhanallah, or exalted is God, and this is the marvelous creation that we would expect from an all-knowing creator, and an all-powerful one, an all-wise one. And it is definitely a fingerprint, a sign that says that belief and faith are not afraid of science. The more you learn, the more you find the evidence you're looking for. It should take you closer to believe that the apparent autonomy of nature is only apparent. It's just because we're used to it. But when we look deep inside or deep into space, whether through the microscope or the telescope, it becomes apparent that the macrostructure and the microstructure of the universe and life and the unseen world can only be there if somebody really intended them to be there and stay there. And I wish to hold on to this idea. So as the finale of this wonderful discussion. So if you have a final note about this,
I think my response would be similar to yours. God is to be exalted.
Yeah, absolutely. So I hope everybody who's watched this is as thankful to the knowledge you've offered us today as I am, and as thankful to the fact that we today live in this world, live in this day, to witness this and to understand this and to tell our children about it, and to really see that the science of today has reached deep enough in the cup or the glass of science, that the initial intoxication that happened a few decades ago, for mysterious reasons, or maybe intentionally, now science can be the sobering effect that takes humanity more towards God and towards belief more than any time else. So thank you, Fuz, for this. This has been quite a blast, and I'm looking forward to the next one.
Yes.
And for everyone who's watched this, please give us your comments and questions, and we might as well be doing an episode where we will address them. And see you again next week. Thanks a lot, Fuz, for this.
Thank you, Ahmed.